Hi, my name is Brian Pettygrove. I'm a PhD candidate with Dr. Phil Stewart here at the Center for Biofilm Engineering. And today I'm going to walk you through a protocol that I commonly use to observe interactions between surface adherent bacteria and host immune cells. Now, this method can be adapted for a couple of different applications. One would be looking at attachment, detachment, and growth of surface adherent bacteria. A second is, like I mentioned, to look at interactions between immune cells and those surface attached bacteria. And then finally, you can also use this method to look at the effect of a surface or a surface treatment on behavior of immune cells. This is the surface that I use for most of my assays. It's really just a petri dish divided into 4 quadrants with a glass cover slip on the bottom. And this facilitates the microscopy using our inverted confocal microscope. First, we start by preparing our bacteria. So I will typically centrifuge a ml of my overnight culture, wash that with PBS, and then perform a dilution down to a specified OD 600. Once we've prepared the bacteria, we will then inoculate the surface. And I typically add just 10 microliters, a very small dose of bacteria to that surface, trying to make sure that we're getting our inoculum in the very center. At this point, we'll then put the dish in the incubator for 30 to 45 minutes to allow those bacteria to adhere to the surface. And leaving in for those longer time points will lead to more attachment of bacteria. All right, once we've allowed the bacteria to adhere in the incubator, we're next going to remove the inoculum, the excess inoculum, by gently pipetting. And then we'll rinse off any non-adherent cells by applying 10 microliters of phosphate buffered saline directly to that spot where the inoculum was sitting. And then we again remove this PBS. All right, once we've rinsed off the cells, we're going to go ahead and add our growth medium. And we'll gently apply this to the edge so as not to disturb any of the adherent cells. For our growth medium, we use 10% human serum diluted in Hank's balanced salt solution. This serves double purpose of acting as a nutrient source for the bacteria and conditions the surface to allow for immune cell attachment and movement, and then opsonizes the bacteria. When we add our immune cells, we add a small quantity directly to the bulk media, and we do this to avoid disturbing the adherent cells. So they will settle down in the matter of minutes, and then you can image interactions between those immune cells and the bacteria on the surface.
Finally, if you would like to image a surface that is opaque, we would propose a scheme like the one depicted here, wherein you place your surface of interest inside of a flow cell that you can then fill with your media of choice, and then you can image the top of that surface using an upright microscope rather than an inverted microscope. With that, thank you for your attention, and please don't hesitate to reach out if you have any questions.